Professor Bates was appointed to a personal chair of individual differences in psychology in 2007, having arrived initially at the university in 2005. He studied and began his academic career in Auckland, New Zealand, and came to us from Macquarie University in Sydney. His research has focused on the genetics of individual differences, and he has a very distinguished international reputation for linking genetic characteristics with aspects of a range of language acquisition, reading, remembering. And one of the characteristics of his work is his interest in, and indeed his exemplary work in, multidisciplinary teams of scholars. These are teams who work across intellectual traditions, but also across institutional and indeed international boundaries. And this is a very important aspect of his work, which has led to very many scientific breakthroughs. I think another characteristic of his work, and one that we'll very much see in his talk this evening, is his interest in explaining the underlying mechanisms accounting for an enormous range of human behaviours. Happiness, memory, language development, and even more that we'll hear about this evening. So I'm delighted to introduce Professor Timothy Bates with his lecture, Psychology in Your World.
Thank you very much. Thank you. Thank you all very much for coming today. I was just saying to Dorothy before, I don't think I've ever been nervous before a talk in my life, and I feel a little sense of butterflies today. And I guess it's a sort of sense of awe at the magnitude of the tasks that we take on, thinking about the world, but also the responsibility we have in an institution like this to be doing interesting things and to be communicating them. And so it's been a great experience putting this together. I'm very glad that you've come today. Appreciate it very much. I wish my mother could be here. She's pretty much as far away as you can get on the other side of the world, and so hopefully we can send her this movie or Twitter her, I'm told. You can imagine being pretty much as old as men could think and consider things, and looking up to the sky and thinking, how does that work, and how do my seasons work? And so there are some very old sciences that have got a much longer tradition than psychology has. We're a very young science, even compared to cognate disciplines, things we're very close to, like medicine here at Edinburgh, that they're coming up to their 300th anniversary, and we've just had our 100th. And both of us needed some consideration and opportunity from society. The medical school needed a society that thought that possibly Burke and Hare stealing bodies was going too far, but that the doctors who were cutting them up and examining them were doing a good thing, not a bad thing. And in psychology, we need to take even more of a liberty, I think, with people's expectations, because we're going to dissect the human mind and things that are often thought of as the human soul, and it really has been quite challenging. And you can see it even now. Many of the topics that we study are amongst the most controversial and challenging, and they evoke the passions as well as challenge our reason. And for that reason, that's the theme I've chosen for the brief time I've got with you, is looking at some of those passions, the things that you can evoke, and reasoning how we think about these things that are evoked in our minds. So what do we do with this liberty, this privilege that we've gotten so recently in the last 100 years? I think often people think of psychology that it's just going to be common sense. You can't tell people anything about a mind. They've already got one. You live inside one every day. And so maybe it looks sometimes like we simply take newspaper headlines a bit too seriously, but many of them are really serious issues. The Royal Society of Chemistry reported this year arguing that teaching standards were risking the nation's future. That's a serious thing. We need to figure out whether we're doing the right thing with teaching or not. There are policy decisions. Is it a good thing for teachers to need a 2:1 degree or not? We can address that. Is that going to help people learn, or is it irrelevant, as we're told sometimes? Very personal decisions. Should you breastfeed your baby or play at Mozart to raise IQ? That's a couple of bits of work. One we've done coming up this year, looking at a gene that might have meant that for some people breastfeeding was very important. Sadly, it doesn't seem so, although that's actually good news because it means that bottle feeding is fine. So we're addressing a whole range of questions which are quite pertinent and often find themselves in the news. One that I'm going to start with, just partly because it's topical and interesting, and partly because it gives me a chance to advertise the research of my very good PhD student, Gary Lewis, who's been working on coalition affiliation. And you can see here these two men who've spent the last, perhaps most of their lives, perhaps certainly recent time, arguing that each other's group is wrong about most things, and now they're working together. And they've had to form a new coalition. This area of coalition psychology, especially known under different names, favoritism, in-group favoritism, goes back to the beginnings of psychology, reference there from 1906. It's another important area. There aren't many headlines that we see in the newspapers that don't have something to do with group and intergroup conflict. So trying to understand this is really something that's quite important for us. So I'm just going to highlight for you a little behaviour genetic model, which I'm going to go into some detail in explaining in some work that another PhD student and I have been doing on happiness. Just to highlight a couple of things, in Gary's research on coalition affiliation, the ability to form a group, this A is for additive genetics and E here for unique environment, the things that make each of us unique, even when we're born with the same genes and growing up in the same family. And you can see here quite large numbers, 0.7 heritability of a mechanism which the modelling allows us to test whether there is this single module or mechanism in the mind that allows us to affiliate across, for instance, caring that people share or don't share our religious beliefs, worrying whether people have the same ethnicity or the same race as us. Three major features around which groups coalesce. So there's significant amount of heritable variance which allows us to tease apart these modules in the mind. We end up then with what we call a dual root model of favoritism. One flexible and general mechanism for coalition formation, and some more primitive essentialist mechanisms which also pull us towards forming groups and make it harder to form groups around different boundaries than we sometimes do. So it's both a very important thing for us to understand, it's a difficult thing for us to cope with, and here's an example from a twin study with something like 1,000 families indicating that we seem to be biologically constructed in such a way that groups have a place to live in our mind, and that different elements of groups have different places to live in our mind. So we can really get to understand what's going on in our heads if we look and if we use some of these tools like twin studies to do that. So I said I was going to structure my talk around two different kinds of things. They go back to a painting that you'll see in the university, and a man who's really central, not so much to the university, but to Edinburgh's place in a privilege that people gained to begin to question things that everybody else thought ought not to be questioned. And in thinking about morality, this is a very reasoned statement. Really, you can spend some time thinking about what Hume is capturing here. That passions are things that can be excited, and that they're not going to be things that reason has a particular potency in. And that therefore we shouldn't look, if we're thinking of rules of morality, to our reason, but think of these as two different things that are going on in our minds, which can talk to each other, which can interact with each other, but which are fundamentally different in terms of their motivations and how we process information. So I thought we'd just start with a pretty, again, a topical and on the face of it lightweight, but really pretty interesting and important to all of us, the built environment. The father of the figurehead of our university, the son of the figurehead of our university, Prince Charles, says modern architecture is rubbish. Could we address that as psychologists? It's a question really about a passion. People think that this is a terrible thing to say, or think, my God, thank God somebody's saying that. It's a Humean question. We're really asking then, can architecture have something in it that arouses a passion inside us? And if so, what would that passion be? If architecture can do that to us, it means that we've got a mechanism in our mind that it speaks to somehow, and that we evaluate, and that we're going to make distinctions between different kinds of structures. That mechanism may be one that can be connected to arbitrary standards. You could grow up learning that any kind of particular building is gorgeous, another one is terrible, and it might be arbitrary in that way. but it might also be that there are key things that a building has to do in order to remain beautiful across times and across cultures, across societies. In which case this native passion might be tunable to detect a range of functional requirements that we see in buildings and that evoke this feeling of beauty in our mind. You often hear the saying, beauty is in the eye of the beholder. Shakespeare did say that a rose by any other name would smell as sweet, but regarding beauty, he speaks through the words of a character in Love's Labour Lost. My beauty, though but mean, needs not the painted flourish of your praise. Beauty is bought by the judgment of the eye, not uttered by base sale of chapmen's tongues. What Shakespeare is saying there is that beauty is not conjured into existence by mere words, and it's probably not then just going to be in the eye of the beholder. It's something that a discerning observer could extract from the environment, and that maybe then there is this system that we've got in our mind for finding something that's beautiful. This is what Carlton Hill used to look like when I was first here in Edinburgh. I would walk down here. You're kind of sitting in John Lewis now looking across. And I thought this was a lovely park connecting Carlton Hill down to the city, but it turns out it was vacant land that the council could dispose of. And the plans went up for a building to go there. And this is the web advertisement for the building that went in its place. They're talking about, the building is called The Cube, and they're talking about what it's going to do and telling you how good it is. And in advertising it, they point to this icon of our city in the park, and it's beautiful, and this is something they think that we'll find attractive. And of course we do. Millions of people come to see what we've got. But that's what they built. And so we have the question then of why, when you're building a modern functional office block, do you advertise in this way? What is it that people are having in their minds that makes them find one beautiful, and therefore that you advertise it, even though you're going to build something different? So we can begin to ask Hume's question. We could also answer Kant's question about what is it that we're built to perceive and how do we do that. But I'm just going to focus in this little brief part of my talk today on wondering whether beautiful buildings make us happy, just one little experiment. So that the way that we work as psychologists is to think, well, this is something that people think must be true, can't be true, could be true, but we can test it. We can ask empirical questions of the world. So we can ask, do people rate buildings reliably? Do they rank them in a particular order? And then we can see what effect they have on us. And finally, then we could go down and explain the mechanisms in the mind. So here's one little experiment with 18 people. We got them to rate buildings, 3 weeks apart, just buildings around George Square. And the retest reliability for those people was very high. So people said the same thing each week. And they might have, of course, been ranking them in very different orders but being very reliable in that. If we had one of these mechanisms that could connect up to anything, people could rank things in arbitrary orders. But a couple of experiments later now, with more people rating these buildings, it turns out that the rank order of buildings is highly reliable across people. And in fact, our ratings of beauty are massively predicted by what it is that we're rating, in a way that you can completely predict whether someone is going to say what's in front of me is attractive by knowing what it is that's in front of them. It's R squared, so 3/4 of the variance was explained by what building you were looking at, which is basically all the variance there is to explain there. So there's something in buildings that people are agreeing on, quite diverse ranges of people. We've looked at students and people inside and outside the university, controlling for a whole range of factors that might influence what you're rating. We've also looked at high-end architecture. This is, if you've been to Los Angeles, you'll recognize the Disney music building. Using, again, houses noted for architectural merit. And these also have very high reliabilities and great control over whether people think that they're attractive or not. So there's something being aroused in people. They're quite happy to say this is beautiful. They don't ask, what do you mean? They know immediately. And we could move on from here, and if the lecture was going off in a different direction, I could give you my answer to Kant's question of why is it that our mind has to see beauty in this world? Perhaps like people, you just need to be tall, thin, and symmetrical, and that will make you beautiful. There's a curmudgeon who goes along with Kant, of course, Arthur Schopenhauer, who would would disagree, because here's a tall, thin building that most people don't think is beautiful. So we're going to have to work to figure out a theory of beauty and what it is actually that you're extracting from those buildings. But we can move on to Hume's question of does beauty evoke well-being? Does it connect to some passion? And it turns out that the answer to that is yes. With a bunch of people, I did an experiment getting them either before they came past an attractive or an unattractive building, or after they'd been past that building, and going both ways on the street. And it turns out that when you go past buildings that other people think are beautiful, your happiness goes up. And when you go past buildings that other people think are unattractive, your happiness goes down. And when you turn around and do it the other way, it still works. And you can control for a whole range of things. You think, well, maybe this will wear off after I've been in Edinburgh for 5 years. I'll adapt to it. But it doesn't seem to be the case. Controlling for how long people lived here, whether they saw the building every day, the purpose of their journey, whether they used the building, that they actually improved the effect of the clarity of the effect on well-being. So it seems like the built world is doing something that people agree on, and it's connecting to a passion. It's making people feel happy if it's beautiful, and less happy if it's not so beautiful. So that raises the question then of what's it doing for us? Psychology can inform us about the origin of passions like that, of feelings of beauty in the world. David Cameron is now going to have a happiness index for Britain, and that will be part of the outcome politically for rating whether policies have been successful or not, whether they've made people happy, which tells us then we really have to know some more about happiness. We're going to spend a lot of money collecting this, and it's going to influence whether we think we've done a good job or not. So what is happiness? What is it that people are doing when they're feeling better after walking past an attractive building? Happiness we internally think of as something that we work to get. We're running, we're expending effort, but we're moving towards the beautiful tropical island with sun and things that we think are good. Research tells us that running is more like running on a treadmill, that it's hard to get advanced, and that if you stop, you've really got to run in order to stand still, which makes us feel at sometimes that we're like Sisyphus. And this is actually more, I think, in this illusion than initially meets the eye. Climbing up the hill with the rock, but every morning the rock is at the bottom of the hill again. We're somewhat like that on our treadmill. There's also an element of adaptation here. People adapt to the burdens that are put on them, be they illness or wealth or poverty, and feel about as happy as they always have. We paper with Alex Weiss, who's here in the audience, and Michelle Luciano, also indicating that happiness is a heritable quality that differs between people. So this view of subjective well-being as hedonic well-being says that it might not be something we can easily change because we've got to work to stand still. But it's only one view of well-being. It turns out that there's a very different view of well-being. I'm lucky enough to have another PhD student, Despina Arkantaki, who's working a Greek name, and it's quite nice that it matches up with Aristotle, who focused on a very different idea of what well-being would be, that it would be optimum activity of the soul, eudaimonia, a good inner being. It would require good character. He thought that you would need to be taught. You would need to be habituated to this work, and that it would require conscious exercise of choice. And so we've got two very different ideas of happiness. Which one are we going to put in the National Happiness Index? How do they relate to each other? I'm just going to give you a brief overview now of a twin study where we try and look at the relationship between these two different kinds of happiness. One, eudaimonic, relating to moral virtue, a characterological trait, challenging probably to achieve, only evaluated at the end of a good life, versus hedonic well-being. It will be lowered if you have to work harder to get the thing that you want. It's robust to life's challenges. We adapt to it and seem to have strong individual differences that are heritable. And it's often associated by contrast with virtue as being base or pleasure-seeking, something not as honourable. Could a twin study help unravel what's in the mind and therefore to understand what happiness is? These are different kinds of twins, each of which is critical for our study. These two girls are identical, monozygotic identical twins. These two young women are very similar looking to your eye, but it's probably just because they're both attractive. They're not identical twins. They share only half of their genes. And the two kids who are down here are with their gorgeous parents are opposite sex twins. And so we've got two people with exactly the same genes, two people sharing half their genes, two people sharing half their genes but differing in their sex. And that allows us to do a whole lot of contrasts about what genes are controlling which behaviours by seeing how they tumble through the generations separately and which traits they go with. A little bit of mathematics for you. You'll often, if you're a Radio 4 listener, whenever people mention twin studies, it's usually in close juxtaposition to, and they have assumptions and you can ignore them. So I'll give you some of the mechanics and what the assumptions are for twin studies. The identical twins share 100% of their genes, and therefore the correlation, this MZR, we can think of as 100% times the effect of genes, because they share 100% of their genes, and 100% of the family effect, because they're growing up in the same family, they've got the same social position, the same postcode, lottery ticket has been bought for them, both of them are going to have the same likelihood of having a bedroom or a bathroom in their house, there'll be books in the corridor for both of them. So that's all shared. The DZ twins share all of that family, but they only share half the genes. And that gives us a very simple set of linear algebra that we can use to try and model then the heritability of behaviour. We can look at the role of genes by subtracting that MZ from the DZ correlation and multiplying it by 2. And the reason for that is simply that if you look at the difference between these two equations, they're identical in sharing family. They only differ in 1 times the genes versus half the genes. So the difference is half the genes. If we take the difference then, we've got half the genes, and if we times that by 2, we've got the full genetic effect. That's the rationale of the twin study. And we can carry on then through, once we've derived that genetic product, to looking at unique environment. It's going to be 1 minus the MZ correlation, the things that make us different by chance, even though we're growing up with the same genes and in the same family. And family environment, we can 1 minus the A and the E that we've figured out. It's what's left that's making people similar in families. What does this tell us then if we know it? The first thing is to see that we can see this as a picture. Anything we're measuring down here, coalition, affiliation, I put up happiness that we're going to look at now. We could have additive genetic effects, family effects, unique environmental effects, and we can decompose our behaviour into these different components. That's less interesting than using this method to try and see whether different parts of behaviour overlap or are distinct from each other. So a quick look now at 2 different kinds of well-being. We can measure subjective well-being, hedonic well-being, by asking how good do you feel and how much pain do you feel. It really is that positive affect and negative affect added up together. So if you've got lots of positive affect, not much negative affect, you're feeling good on average, there aren't many things causing you pain, you're hedonically high on well-being. Eudaimonic well-being has got many more facets: acceptance of life, but autonomy, mastery of your environment, and yet a sense of personal growth, a sense of purpose in life, and a feeling of warm, positive relationships with people. As Aristotle in part thought of it. There are some things that in his model that are missing there, but also people like Maslow and self-actualization. It captures very similar concepts. And we can ask people about these and assess them. And when we run this model, what we find is that this eudaimonic Aristotelian well-being, the characterological virtue, is quite heritable. There are some people who've got more of it than others for reasons that appear to be genetic. And that reaches out to hedonic well-being. People who are high on eudaimonic well-being also seem to be feeling pretty good and less often feeling bad. So if we raise eudaimonic well-being, that would suggest that we might also be raising people's hedonic pleasure. And you can see here a second set of genes that go off uniquely explaining a sense of pleasure independent of living well. And so these are distinct concepts. And it will matter for us quite a bit which of these we put into our national index. If we focus on hedonic pleasure seeking, we can move in one direction. If we focus on eudaimonic well-being, we can move in another direction. So it's important to understand how these 2 different concepts fall apart in the mind. So we can study things that we like, that we dislike, like buildings, or and we can see how these make us feel good in different ways, whether they make us smile or whether they make us feel like we have a sense of purpose in our life. But we can also ask questions about the second big part of mental life that Hume distinguished, which is our reasons and our reasoning ability distinct from our passions. It's going to look at a couple of things. How does reasoning develop, and upon what does good reasoning depend? I'm just going to focus on almost entirely on, in the one case, on intelligence and relationship to developmental challenges, looking at bodily symmetry, and then work that I've done with Michelle over the last 5 years in the genes for reading and language. So, I find this a quite captivating image. The newborn baby's hand, tiny as you can see, just grasping around what is clearly an old person's, perhaps their great-grandmother's hand. The lifespan development is captured in there. These are different in all sorts of ways, in skin quality, and one has lived a full life. What is it that is present, perhaps even as this baby is being born, or maybe shortly after, or during early childhood development, that predicts things about aging? We're interested in how long we live and how we can live healthy lives. So we want to study links between early development and late development, and continuity, and any innovations that are happening in there. One way to think about development, because we're made of trillions of cells, is that the job of development is to assemble those trillions of cells in the right places at the right time. And if you think about it, the decisions that are being made are microscopic and manifold, and it's almost unbelievable when you look at the tiny scale that these biochemical decisions are being made on, that we can reliably develop at all. I've got a little picture here which will come up later in the work that we've been doing in dyslexia. These are cortical cells migrating across the neuropil, building a cortex. This is normal development. The cells start off at the bottom and spread out, building a cortical layered structure. You can see here knocking down some genes that are important for that. All of this is occurring across a tiny little piece of space in which millions of cells are making decisions about how far to migrate, sending out axons, deciding who to talk to. So there's an incredible amount of microstructure, which precision is going to be really important about getting it to work properly. A major figure in Edinburgh's biology department down at KB, Waddington, back in 1957, focused on differences between organisms that would impact on our ability to build this system, this complex system, reliably so that it functioned well. And he captured this in the idea of canalization, that evolution would tend to encourage the development of scaffolding and redundancy, which could support and back up development in such ways that made it reliable, so that we could reliably avoid noise and achieve successful development. Development then in part reflects genetic differences. That Waddington really made famous this diagram of a kind of an environment down which cells can, these are single cells deciding whether to go this way and become one kind of cell, or as the stem cell differentiates and becomes a different cell. If it makes wrong choices, it's going to find itself very hard to get over these humps which are canalizing that development. So you've got a series of choices to make about what kind of cell to become, and a lot of control to exert to get each cell to become the right kind of functioning differentiated cell at the right time and in the right place. And what that means is that things like symmetry, where two fingers have grown up to be the same, if you look at your own hands now, you'll see that by and large these two fingers that have never seen each other, didn't talk to each other, were not measured against each other, have done the same developmental plan. This is work that a third very good PhD student that Ian and I are lucky enough to have now has been really advancing studies in over 4 or 5 different fields pretty rapidly in a thing called fluctuating asymmetry, where if this program is highly canalized and is able to develop, to canalize development to meet the achieved goal, then a side effect of that will be that bilateral body parts will also end up being symmetrical, because both of them have been canalized to the same goal, both of them will achieve the same development. And so we can use symmetry as an indication of differences in the precision of this developmental blueprint. So I said you can hold up your hands. There's ideas going back before the last war and more recently of using fluctuating asymmetry to measure this concept of developmental precision. It's a distressingly difficult concept to get your head around. Fluctuating asymmetry doesn't fluctuate, although it does change very slowly, and it's really about symmetry, not asymmetry. So it's almost completely wrongly named, and if you're trying to explain it to someone. But what we can do is go around the body measuring lengths of fingers and of diameters of wrists, heights of earlobes, facial symmetry, a whole range of places where we can get some index of how perturbed development has been very early on and later on. And we can summarize all of that as an index of developmental disturbance, or if we think about it the other way, the ability to canalize development with high precision. Some work I did actually starting before I came here, built on work of others like Prof and Miller, demonstrating that IQ is related to symmetry in adolescence. There's a couple of studies with I think 300 people in them, between them. It's reasonably reliably found that symmetrical adolescents do better on reasoning tasks. Why would that be? Well, if we think of development as being this goal of getting trillions of things done at the right place at the right time, one place where that's going to be most critical to get right is in the brain, which is our most complex organ, with trillions of cells going on that all have to be organized to work correctly. So we can think then of bodily asymmetry as being an index of development that's been perturbed, and we would expect therefore to find a negative impact on cognition, because it's going to be very complex and really critical and dependent on getting the physical organization of the brain accurately in place. So if we move now to a study looking across a much longer period of time, we can begin to get a feeling for what the connection of symmetry is across multiple decades. And we're lucky enough here to be able to collaborate with the Case Centre and Ian Deary, who's in the audience, and the Lothian birth cohort. These are all people who were born in 1921. They've been studied since 1932, when they were aged 11, and they've been followed up at age 79, at age 83, and at age 87, and soon age 90. So you can see here reaching across 3/4 of a century, and we can look at how their fluctuating asymmetry that we can measure relates to their ability, their cognitive ability, over great big periods of time, 3/4 of a century, which is about as big as it gets for human beings. And here's a table which I think doesn't necessarily convey as intuitively the results. So I'll just switch to a graph. And what we've done is used fluctuating asymmetry measured in these 86-year-olds, and we've related it to their current cognitive function. And you can see up here that fluctuating asymmetry in relationship to their current cognition, the relationship is about, effect size is about 0.2. If we go back through the 3 waves that they've had recently, you can see that those relationships are all clustered around 0.2 and neither going up nor down. But the interesting thing, I think, is in addition to that, is not only that being symmetrical in your 80s means that your brain is working reasonably well, and you can think of different ways that might come about, resisting recent challenges or beginning to age more rapidly. But if we look all the way back through those decades to when they were 11, the relationship is just as strong as it is across a week or two. And so there's something which symmetry is tapping into, which was already doing whatever it was doing in terms of mental function 3/4 of a century earlier. So this gives us a clue that something about development, about organizing those neurons, is going to be very important for implementing the functions of the mind, the ability to reason, and we've now got an index of how well that person was put together, and we can see if it influences how well they age and how long they live, and a whole range of other things. It also allows us to look at the kinds of things that might challenge development. This is the beginnings of some work that we're doing, looking at the effect sizes of age 11 and age 87 ability on related to fluctuating asymmetry. And again, this is not a compelling presentation in a table, but what it's saying is that we can control for a range of things like paternal education and height, and David's been doing some much more complex work now controlling for a latent indicator of resources present in childhood, and certainly for bodily asymmetry, not really reduce the size of the relationships at all. So it looks like something that was either present in the womb or in the person's genes, but isn't as simple as how many bathrooms that you had, is building this relationship in. So it begins to let us point towards different causes for development and the canalization of development. So, so we can measure something as crude as the body and find results across periods of time as great as 76 years and counting, hopefully for as many people of these people as possible. We can begin to look for causes for those relationships, developmental resources or status, which don't really seem to be driving it at the moment. But so then we can begin to look at molecular mechanisms, and that's what I'm going to turn to now, focusing on language and reading. If we summarize that idea, that a lot of development can be viewed the same way that Heston Blumenthal views molecular gastronomy, cooking, as a case of precision being actually what makes the recipe. Here he is measuring things out precisely, and that in human development the same sorts of processes are really important, that getting things exactly right is a lot of what makes that recipe work, that there's a kind of developmental molecular gastronomy in building a person who functions really well in that Humean sense of reasoning about. There's a whole range of reasons why you can think that is going to be particularly important in the brain. It's full of tiny little, this is zoomed in on a single synapse, and even when we get to the point of having gone through billions of neurons down to one particular component of a neuron, its synapse, you still find dozens of parts. It's more complex by orders of magnitude than any machine we've ever built. And the levels of precision in these kind of synaptic alignments and the relationships that proteins have to have with one another in order to work effectively are truly on the atomic level, at a point where precision is almost theoretically hard to gain, let alone practically doable in a machine as vulnerable as we are to the slings and arrows of outrageous fortune. There are a range of, this is a little study from autism where people are looking at how proteins can work together to align cells that they can talk to each other, and increases the precision with which they can do their computations by increasing the precision with which they're assembled. And this scales up also into columns, and indeed once we reach the size of the whole brain, if you're going to build a bigger brain, then the distances across which you're connecting things become larger, and errors become things that would have left you still connecting to the right place mean that you're now off target because the distances are so much bigger. So there are a whole range of challenges that humans uniquely face with such big brains that are so important to our way of life. And we've begun to unpick them with the molecular genetics of neuropsychological development. If we just cast back to a headline, this is one that comes up routinely, that dyslexia doesn't exist. In this case, who is it? Julian Elliott from Durham says this is just a middle-class way of hiding stupidity, he says. Well, if that's true, then really that's a claim that we're a box that doesn't have a place for reading, or perhaps a box that doesn't have a place for language, but we can study that genetically. And that's what we've been doing for the last 6 or 7 years. Communication disorders are in fact common, even when they're not being used by students to ask for different treatment. They're in fact real disorders. Dyslexia is a failure of print to speech conversion, of reading to speaking. But we also have language impairment, which is reasonably common. Also, we can see it in agrammaticisms, but also a whole range of other anomalies in spoken language, deficits in semantics and syntax. So there's a large group of, especially children, who are affected in these ways, and we can study that. I don't know if this is going to work or not.
Assess not only the health of the crops and the food, but also the potential for contamination, the commercial implications for Australian primary industries, and the benefits and risks associated with genetically modified organisms.
Okay. So before I came here, I was working in Australia for 10 years, and this is a member of the current Labor government demonstrating for us either language or reading problems. So they're fairly real, and they affect adults as well as children. But you can still obviously be quite successful and have a problem with language. So there's been a bunch of revolutions in our understanding of communication, especially around these two guys, Jim Watson, who's still active and bouncing around, and Crick, who's now dead, in uncovering the secret of life in DNA. And that's really helped us then begin to look at the molecular genetics of communication. One of those jobs that we're going to have a lot of precision in getting right. We want to find out then what kinds of precision is it, what connections are we building in the brain. I was working with Max Coltart before I came here down at Macquarie University. He's most well associated with this dual root model of reading, where letters come in, we've got a machine for extracting what letters are being placed in front of us. We've got a root that we can use to pronounce words that we've never seen before, which is a great aid in teaching yourself reading, because you don't have to know what a word is. You can sound it out, match it up to something that's already in your lexicon, and then and know what it means. You also have, though, an orthographic lexicon and a phonological lexicon, so a box of words based on their orthography and the sounds that those same words should make. And both of these routes allow us to get from a written word like dyslexia down this route to dyslexia, and down this route to dyslexia. There are different models of reading. All of them have at least two of these roots in them, though they might characterize what they do differently. They make very similar predictions. So we've got a set of behaviors to understand. So we're studying genes for reading. When I signed the big book to become a professor, there were two language professors who were present at the same time. And they said, "What do you do?" And I said, "Oh, we're doing genetics of reading." And they said, "Well, that's impossible, isn't it?" So I thought, that's good. Do two impossible things before breakfast each day. Well, is it impossible? This is a paper that Michelle and Nick Martin and Margie Wright and I did in Queensland Institute of Medical Research. We're looking at irregular word reading, words like yacht that you can't sound out, non-word reading, reading things like slint where you have to sound them out. So two different kinds of reading. And it could be that it is impossible for these to be heritable, that there's no genes involved. And we found a different sort of answer. You can see here some quite large numbers. High heritability, these are additive genetic influences on irregular reading, irregular spelling, non-word reading, non-word spelling. The non-word spelling slightly less, but that's mostly because it's a much less reliable test that I was responsible for. But a highly heritable disorder. And the other thing that's interesting on this diagram is that it demonstrates that there are two genetically distinct forms of reading problem. These genes can't be dropped without reducing the fit of the model. And what they're saying is that some people have a particular problem with building up a sight vocabulary of words and knowing how they sound and using them. And this is also where we get from words to meaning. And over here, another set of genes which are required to explain non-word reading, that grapheme-phoneme conversion system. So there are two genetically distinct forms of dyslexia with lots of shared components, because of course there are shared components on the input and the output sides of this model. So it does look as if there are genes for reading. But that comes from a twin study, and I've tried to give you the mathematics for it and convince you that it was exciting and valid and not flawed by associating it with a nice warm concept like eudaimonia and character. But often when twin studies come up and say that something is genetically influenced, people think, well, those are flawed. We can avoid those, can't you? Development is so very complex that we could never find these genes. This is just going to be a fundamentally difficult task. If not, we've moved from impossible to just extremely difficult. And we had quite a lot of success early on. People studying severe cases of dyslexia, so these are mostly samples where people have recruited on the basis of severe problems with reading and some of them for language, have found a dozen regions of the human genome linked to reading problems. So it looks like it's not going to be so difficult to find some of these regions. And the response to that, of course, is that, well, maybe in these very severe cases, people really with severe challenges in learning to reading, that will be genetic. But those would be more like mutations, perhaps, rather than normal variants in reading. And that's quite possible. And it's quite possible that normal variation in reading would reflect purely access to adequate education. But already we'd shown a highly heritable form of reading in our unselected Queensland sample. There well over 1,000 families, normal children. And it was quite heritable. And so it's not surprising that when we look in our sample using linkage studies, we find the same genes, chromosome 15, chromosome 18, there's a group down in Oxford have been studying chromosome 7, overlapping with the severe cases of dyslexia. So it really looks like normal variation in learning to read is a complex genetic system. It's distributed across a whole range of chromosomes, but the same things that are going wrong in severe cases are going wrong in normally varying reading. So these look like little blips, don't they? I haven't even had time to tell you what linkage is, and we're going to move on now to gene finding. People might think, well, how is this going to explain children at school? Because it's not really compelling until you get particular genes, I think, where just like that photograph of astronauts looking down on the Earth tells you we really are a planet floating around in space, finding a gene that does something in the brain kind of tells you what the mechanism is and really is compelling evidence rather than statistical evidence. And so it's an important challenge then to move on to find genes, because we could be wrong, there might be some artifact driving this, and it would also be great to find a theory of what is it that is trying to develop to build a human being that's got a language instinct and the ability to read. What are the special tasks that are being done in the mind? And so we've done that in a series of papers. This in a biological psychology, we're exploring a gene called KIAA0139. KIAA means don't really know what it does yet, but it turns out we do know a little bit about what it does. When you knock this gene down, it mucks up cortical migration across the cortex. So it's a gene that's involved in one of those precision decisions that a cell has to make. A cell is sitting there in its little stem cell form, rapidly let's make 2 stem cells, and then it's also doing differentiation to try and become some final product, and it's also migrating through space to decide where's my cell body going to be and who am I going to connect to. All decisions that take place in space and time, and where you make that decision will determine where you are and who you connect to. So there's one gene which is implicated in reading, this one's on chromosome 6, which has something to do with cell migration. And here's another gene. This is a paper with a co-worker, Penelope Lind, and again our Queensland colleagues published in the European Journal of Human Genetics, a double cortin-containing gene. We have a look down here. When we knock it down, you can see that the cells get off that base layer, but then most of them clump and there's much less differentiation through the cortical neuropil. These cells are getting a signal to stop too early. They know kind of what they're supposed to do, but statistically many of them are putting the brakes on too early. We can carry on and look at some other genes. This is a recent paper in molecular psychiatry on a gene DYX1C1. It's DYX1C1 because it was the first candidate for dyslexia that was reported. It was actually the first ever linkage to a cognitive trait, Smith back in 1983. So it's a recent field, but really brought up to speed now as we begin to see, again, this DYX1C1. If we do a knockout mouse in this case, again, we alter the decisions that neurons are making as they distribute themselves across the cortex. So all 3 of these genes are involved in reading. All 3 of them alter the decisions that cells make about migrating up and stopping, 2 different decisions influenced by receiving chemical information about where you are in the neuropil and what time in development you're at. So we even have the beginnings there of a theory that the critical thing that goes on with reading, people who have struggled with reading have got different levels of neural connectivity. Their cells have made different decisions about how many different other cells they're going to talk to, and some specific genes. And we've moved on most recently, just actually coming out a couple of months ago, to be looking at genes for language as opposed to reading. children are fluent speakers. We often think of reading as being a difficult thing to learn. You've got to be maybe taught it, whereas language, infants will invent language. They are often more so systematic than the language to which they've been exposed. There are resemblances across language, all of which makes us think that there's something about a machine that's being built in the head which is extracting regularities, and if it's not finding them, it's making rules about them, and building a language machine in the infant's head, extracting grammatical principles for which it doesn't have evidence and testing them against the language environment that it's finding, which leads some people like Chomsky to think that we have this universal grammar which is being reflected in these behaviours, and more recently people like Pinker and Bloom arguing that this will be an evolved behaviour. It's not going to come about without selection. So what is it that would have evolved to give human beings language? The language system is incredibly complex. We spoke about reading. Language is a much bigger bag of fish. We've got speech sound analysis equipment connected to auditory cortex. We've also got a little device called the phonological loop, and that's what I'm going to focus on for a couple of minutes now, which allows you to hear a sound stream like he, fa, ha, la, ga, and even though that means nothing to you, to be able to repeat it back. And it turns out that in neuropsychological patients who are unable to do that, where the phonological loop is damaged, they do fine on intelligence tests, but if you try and teach them a new language, they are at a great deficit. So it looks like, and Alan Badley argued that this might be a human adaptation to facilitate the acquisition of language. It's a place where you can store words while they've got no meaning, the first time you hear them, as you then associate what is it that the person is pointing to, touching, interacting with, what concept are they describing, to allow you to acquire this word. After you've acquired it, it turns out it's a bit of an appendix. It's not needed for carrying on doing processing, but it's a critical component of language acquisition. And just recently we've studied a gene called ROBO1. It's been associated in linkage regions that are associated with speech sound disorder, which is a little bit like specific language impairment. We also found it, and others have. Simon Fisher's group is now running a Max Planck Institute, and Nopala Hemi, a Finnish group, associated with reading problems. There's obviously some connection between reading and language. And we've recently got evidence for SNPs in this gene. This is not going to mean anything to you if you haven't done association studies, but looking along the gene, this is SNPs, which are tiny pieces of varying genetic material in the gene ROBO1. We have this massive peak. It's a p-value of 10 to the minus 6, associating SNPs in ROBO1 with language. So what do we find when people have a less efficient version of ROBO1? They can't repeat back those meaningless words that you tell them. It turns out this is a normally varying trait, and ROBO1 is doing something that is building this buffer that allows you to have sound going in and staying there over time, and then somehow building like a little piece of tape, half a second long or a quarter of a second long or a tenth of a second long. And what this ROBO1 polymorphism is doing is altering how long that piece of tape is and how durable it is, and making some kind of bucket brigade that keeps that sound alive while you figure out what it means. And it turns out when we're looking then at all the different parts of the brain and how they fit together to make language work, that whereas those reading genes were associated with very small differences in cortical migration, ROBO1 is associated with axonal migration rather than cell bodies over big distances as they cross from the left to the right hemisphere, so making big decisions about wiring the brain up, and also making much smaller microarchitectural decisions about, as axons are moving, making decisions about which direction they travel in. So it works with a bunch of other genes which also turn out to be associated with language and dyslexia, SLIT and NTRK. So psychology is going to become a lot more molecular and a lot more complex as we begin to figure this out. This is rapidly becoming biology, but it's where the answer lies to how our brains are able to, how you're able to hear what I'm saying and understand it, whether it arouses your passions or not. It seemed to have aroused mine early on in life. I wrote a paper for a journal called Mind, Brain and Education, where in their initial issue they bunch of guys at Harvard talking about how they wanted to bring together the mind and the brain, studies of those two things, and how they would relate to education. And that a comment that they often had from colleagues was, "That's a great journal, but can't you leave out the brain?" And so I've just kind of expanded the title a little bit here: Mind, Brain, Education and Society. I think it's just bigger than school. Can't you leave out the brain? And I think that what we know about our origins, about differences between us, about our genome, about our passions, and about our reason mean that no, we can't. That it's going to be essential for us understanding ourselves and fixing some of the problems that we've got to keep the brain in there and to carry on understanding it as we're doing here. So I'm very glad to be here at a place that I can do that so readily with so many lovely colleagues to collaborate with. And I'll leave it there and take any questions if you have any. Thank you very much.
Oh, thanks very much, Tim. That was an incredibly fascinating and wide-ranging lecture, and I'm sure we have got time for a few questions before we go off to our reception. So if you'd like to put your hand up if you have a question, and then wait for the microphone to come to you. So.
I'm just puzzled why the brain picture you are showing is showing quite a pronounced frontal atrophy. Does it have some deep meaning that, in a way, we should
I don't know.
dwell upon?
It could be prescient. It's not mine.
Anyone else? I could ask something about the Aristotelian well-being research. Is there independently of Aristotle, is there a definition of well-being that psychology works with, or do they follow Aristotle's conception of it?
So the question is there do we follow Aristotle, or have people developed their own definitions of well-being? And the answer is that people. I think research went in two different directions with well-being. A lot of people focused heavily on subjective well-being because it really seemed to get a lot of energy and research output behind it, and so that kind of captured the mind space for how people think about subjective well-being, being that hedonic pleasure pain balance. And there's a smaller but pretty robust group of people saying that there's more to well-being than that. Another one's focusing on eudaimonia, and I think it's an active research area. So typically we don't have definitions that we accept and then work with. We are changing our definitions as we're doing the work, and in reciprocity with that. And so that list, Aristotle probably wouldn't be very happy, because there are some things like courage that are not there, because people weren't thinking that courage would be a trait that you should have. And probably psychologists, and including myself, are not expert enough about what Aristotle thought, and do not come across to philosophy often enough to find out and expand our minds in terms of the things that we could be measuring. But then there are other concepts that are really influenced by people like Maslow, and that's sort of self-actualization, and a pyramid, and hierarchies of needs and motivations.
A sort of general genetics question, really, I suppose. I mean, how often in this is a gene really critical, rather than it just leads to a predisposition for something? And I'm wondering if there's more like an analogy, in the sense that your genetic makeup's maybe like your brain, and that the brain's pretty plastic, and so sometimes you can have a bit that's wrong and it doesn't really matter, and in others there's a more sort of critical component in it.
So that's a good question. How do genes influence traits? Do they have great big effects, or do they determine things? Or and the answer is, as often in psychology, both of those things are true. Nearly all, for many of these genes, if they have mutations that break them, that can be lethal or completely ruin function. And a good example of a single gene which has a severe effect on language is FOXP2. There's a family transmitting that who have severe agrammatism, so there's a big effect with one gene. So they can all, when they're not present at all, make a big difference. But for nearly every trait that we study, there are dozens, and for things like intelligence, probably thousands of genes that are all playing a small role. Many of them can substitute for each other, and lots of them have got very small effects usually. So,
so some are highly specialized and some are more general, if you like, or multifunctional, or
So there are genes that are specialized. So for instance, most of those dyslexia genes don't seem to have anything much to do with general reasoning. And actually we partial out reasoning in order to highlight the reading specific signals. So some of them are doing very cognitively specific jobs. Others of them seem to be involved in many different traits. But I think the biggest challenge that people have in finding genes at the moment is just that there are so many of them that usually contribute to a trait.
It's lovely to talk, Tim. Thanks.
I
just wanted to challenge you on one perhaps throwaway remark when you're talking about the phonological loop, and you sort of mentioned that it's important for language acquisition, but thereafter became sort of an appendix. And I wondered if you really meant that, and whether if somebody had their phonological loop removed, whether it would have any impact on their cognitive functioning, and given that there's evidence to suggest it might be involved in some kind of speech input buffer, speech output buffer, involved in remembering serial order, and so on and so forth.
Yep, that's an excellent question. So I was probably trying to protect myself against the opposite criticism, which is that there's some evidence that children with reduced phonological buffer size can acquire as much language as people with a longer phonological buffer once they're adults. Data coming out of ELSPAC with Sue Gathercole. So you're exactly right that, I mean, there's an obvious problem. If you didn't have one, you'd get a 0 score on our test, and then you have to think, well, how much of life is going to be testing that same function? And you're exactly right. Every time that you've got to shift attention and listen to something that was there, it'll be captured in the phonological buffer. So it was a what's the word? It was a throwaway remark designed to protect me.
Tim, hi. Thanks for the talk. When you, at the end, mentioned the mind, brain, and education, you sort of supported the idea that brain is relevant for this endeavor. I wonder if you can speculate how much prescriptive the knowledge of brain can be in informing educational program, or whether science should be descriptive and educational program can develop on their own, not being informed by brain discoveries?
Gosh, that's a great question. That's the sort of thing be the subject of a whole book, I would imagine. I think I have a couple of thoughts about that. I think in many ways the biggest thing that knowledge about the brain can tell people in education is what's reasonable and what's unreasonable to expect about children in general and about children in particular. And so often people want no child to be left behind, or no child to be less than average at a particular task. And I think that one thing that knowledge of the brain can do, and especially knowledges of differences between brains of different children can do, is to let people in education know that firstly that if not every child becomes Albert Einstein, that's not necessarily the result of bad teaching, that it might be also have something to do with what the child brought to the classroom, and that really needs to be taken into account. And then also that we can, if we accept that the children are actually bringing something different to the classroom, then I think that changes how we think about things like setting and trying to target what people are being taught to the kinds of things that children are ready to learn. We already do that on a grand scale. We don't send children to school at 1. We send them to school at 5, and then we don't mix them up with. We don't say, oh well, you're 5 now, you can go to any old class. Let's start you at 15. We do just like ski school does. They say do a little bit of skiing, except then we say, but you can only stay in your own age range mostly. We don't allow much differentiation between classes. And so my guess is that if people saw just how different brains are amongst different children, that they might be more willing to accept a lot more individualized, focused teaching for people, so that it's designed to be learnable by those children at the time, at the mental age that they're at.
Hi, just following slightly from Bob's comments there. Another possible use of this sort of phonological loop type thing is for self-monitoring during speaking. In other words, for noticing and identifying when you yourself are making kind of errors of one kind or another. Now, and also what that suggests, if that kind of thing is right, then it suggests that you're making use of phonological processing very rapidly as you're speaking. One of the things I thought was really fascinating observation you made was you were suggesting that the sort of size of this kind of loop or buffer would be related to the length of connectivity across long distances in the brain. If so, have you got a sort of a way of working out the connectivity of, is there a potential route to working out the sort of connectivity within the brain for language, which is independent of the usual routes of doing lots of neuroimaging, essentially?
Oh, I see. So I was just about to say, you're sitting right next to the person to talk to about looking at connectivity in the mind. So the first thing I thought as you were talking is there are obviously going to be links to things like perhaps if it's self-monitoring, to things like stammering, and there's a whole range of projects that I think can build on the kinds of data that we've collected to go and look at exactly that kind of differentiation of what tasks are and which are not related genetically, and then which genes are driving them. And I don't know that I would turn away from something like neuroimaging. It would be fascinating to look at, and this is one thing that's going on down in Queensland now. We've got several hundred brains scanned, and being able to begin to look at the kinds of differences in brain structure that the genes are making. In addition to the sort of mouse models that I put up, where you can really get down to a single cell kind of level. But I don't know that you can't really work out the function of the gene from the changes that are made to see what it's going to do in the brain as yet. So there's no way of avoiding that step of looking at what it's doing and having that inform you about what these changes are doing. For Robo1, there's a couple of things that it can do, because it's making, one thing it does is in association with SLIT, it sniffs out chemical barriers when it's come up against a midline, which would normally turn around at. And when Robo1 is activated, it will push on through that and carry on and connect up across different dimensions. So what we'd expect here is that there will be more diverse regions being connected when Robo1 is in its active form versus this broken form.
Do you have any thoughts about the implications of this for students with dyslexia and for the help which the university offers to them?
Yeah. So for many of you in the audience, you probably don't remember learning to read, and some of you will have been a struggle that you remember all too well. How difficult it was to acquire, with a massive difference in how easy it is to learn to read. But for everybody, what it involves is being presented with letters and then combinations of letters and words and practicing those. And so just the same things that everybody uses, it turns out, to learn to read are great for people with adolescents with dyslexia. I'm not sure, I mean, I guess what we do mostly now is give people extra time to cope with the reading deficit rather than practice, but phonics and then associations of images with words are two things that practice still does help with. The problem is, as you get older, you may recognize the brain becomes less plastic if it ever was plastic. And so one thing we should be doing is making sure that a lot more of these things get a lot more attention before they come to us.
Okay, I think we should call a halt and move on to the reception. But once again, just to thank you very much for a really interesting lecture and some interesting answers to questions. Okay, thank you very much.
Thank you very much.
This production is copyright the University of Edinburgh.